Okay, are there any questions? Can everyone hear me okay?
Yes.
All right, great. This isn't finishing. Let me start by saying that I almost prematurely closed the lecture. If you were one of the students who left, I did continue lecturing after for about 10 minutes after I said I was going to end. I was mistakenly thinking that the class ended at 6:30, and it doesn't end at 6:30. It ends at 7. Is that right? 6:50. And then the lab begins at 7, and I was thinking it begins at 6:30. I'm sorry about that. It's the regular schedule during the rest of the year, and I was following that regular schedule. Fortunately, a student corrected me, and I realized that we still had class for, I don't know, 50 more minutes. And I decided that I was going to lecture for another 10 minutes. So if you missed part of the lecture, then you'll need to view it online. And I just posted it online. That's why it's a little bit late getting started here. In fact, I just finished now. It may not be available for you to view because it needs to add subtitles to it, but it should be available at least in the next 30 minutes. It looks like it's even available now. Okay. So if you did miss the last 10 minutes of the lecture, you'll need to see that online on my YouTube website. Any questions about any of that? All right, somebody's got a microphone open. I'm going to shut that. It looks like they shut it. All right. All right, we're recording, so everything's good. There's no questions. Let's begin and get started. Like somebody's adding late. Let me state that we almost closed the lecture down early. And if you were one of the persons who left the lecture when I said we're done, please look online on my YouTube website. We did continue the lecture until 6:30. And so we went 10 more minutes. Any question about any of that? Let me state that I did get lab 00 and lab 01 graded. They're both worth 2.5 points. You either got a 5 for the grade or a 0.01 points. 0.01 points means that you can submit corrections by this Saturday. And I think you can submit corrections, yeah, by 11:59 p.m. this Saturday. And just submit another document. This takes unheard of numbers of documents. Apparently people are a little bit confused because I've got two labs on the same, I don't know, thing. And that's because initially this was together as two labs and I decided to break it apart. And that way you have two submissions. If you didn't get it submitted on time, you still can get it turned in by 11:59 p.m. this Saturday. It's just that instead of having two possible submissions, you'll now only have one possible submission. And you'll have no error check on your answers. But you still can submit by this Saturday. Any questions about the lab and the grading of the lab? Let me state, I mentioned in the lecture, if you get 0.01, that is not your grade. That is a code for me on the first grading. And that code tells me to check that lab and see if you submit another, well, corrections, and then go and correct your corrections. So that's not your grade. That's a code for me to know I need to go in there and regrade it a second time. If you do not make corrections, then I will grade your first corrections, which are already graded, and I'll just give you a score based on your first submission. Any questions about any of that? All right, so with the lab, you are allowed to submit it twice. And I consider the lab a learning exercise, and that's why you're allowed to do this. Any question about any of that? All right, if we look on the schedule today, we're going to be doing lab 2, aseptic transfers. And then on Thursday, we'll do lab 3 on microscope basics. You should be reading the textbook on chapter 3 through the end of the section on compound light microscopy. You should do that before you begin labs 3 and 4. Any question about any of that? We'll also discuss and begin the infectious disease project. And I guess I should discuss that now, because I don't have a time for that. If you come down to week 2 of the lab, can you guys see my screen? All right, I'm not hearing any answers, so I'm assuming you can hear my screen.
Yeah, we can see it.
There's a link for infectious disease research project. This is a 100-point project, assuming that you got 100% on your plagiarism tutorial quiz. Click on that link and it'll give you the infectious disease project, which I guess I'm just gonna, actually let me download it. It'd be better to do. This project is like a slideshow presentation. You're not going to be presenting, but you need to send in a report. And it's about an infectious disease. You're going to study one infectious disease and then write a report about it. So you will become an expert on one infectious disease. In this project, you're going to find the cause of the disease, what the microbe does to the human body, how we treat it, social, economic, and political factors that affect our ability to deal with the disease. Let's go on to the project format. You can read about the rationale if you want. Pick an infectious disease and then check with me about your disease and make sure that I okay it. Obviously, I want each student to have a different disease. So the first person who sends me an email in Canvas about what their disease is, as long as it's an infectious disease, I'll write back and say, you can do your report on this project. So email me what your project is. And if no one has your disease, you'll get it as long as it's an infectious disease. You'll need to do a search of the literature to learn about the assigned disease and then write your report. So each person will have a separate disease, and there are ways to make it so that more than one person can have the same disease. Like sometimes people want pneumonia, and I will tell them you can't take pneumonia because it's caused by many infectious disease or bacteria, And so pick one bacteria that causes, or one mold, or one yeast, or one virus that causes pneumonia, and then do your report on that one infectious agent. And so, like I said, more than one person can have pneumonia as long as you're picking different infective agents. Any question about any of that part? And the first one who asks for it will get it, assuming it's an infectious disease. And then occasionally people submit something that isn't an infectious disease. And I'll write back and tell them, sorry, this report has to be on an infectious disease. You can't do your report on something like that. Rarely does that happen. Like once somebody wanted to do it on cancer, and I wrote back, that's not an infectious disease. There are a few cancers which are caused by microbes, but then you'd have to do it specifically on that one cancer. And for the life of me, I can't think of what that is, but I can remember a plant cancer that's caused by a microbe. And then there's a cervical cancer, which is caused by human papillomavirus. So you could do a project on something like that. But if it's not an infectious disease, I'll tell you, no, you can't do it on that. So you need to clear it with me. Any questions about any of that? All right. So when you write your report, you need to give the details about the information of your disease. It should be typed. It should be single spaced, 1-inch margins on each side. And I will require that it be 12-point font or larger. Excuse me. I'm getting a frog in my throat. If you have smaller font, you notice I'm wearing glasses, I'll have a hard time reading it. So I won't like reading it, and I will mark you down. So please make sure it's a 12-point font, and that'll ease my ability to read it. The written report must include the following items. You must have a cover that has the title of your name, the name of the disease, the date, the course name. I do not want a cover page. Just put your cover information on the top of the page containing the first page of your text. Okay? Any question about any of that? I'm just trying to save some computer space. It really was important when I was printing these out and then handing it back printed form, but I don't print it out now, and it's not as important, but still you'll be graded down if you have a cover page. So make sure you just have a cover. And then in the text, it's the main body of your information. It should not exceed 5 single pages. This is only the text, meaning I won't count the cover information. I won't count the references. And if you have any pictures, I won't count the pictures. Okay? In the text, you should describe all aspects of the disease and itemize it and organize it in the report content section, which I will discuss in just a minute. The material should be presented in your own words, and it'll be worth 60 points. There will be an additional 15 points for grammar, spelling, flow, and readability. So if you do a good job, this will be worth 75 points. As much as possible, the text should be written in your own words. If you do not put it, meaning you use somebody else's words, and do not put it in quotes and say where you got it from, you will be marked down, and you'll be potentially marked down to a 0 because that will be plagiarizing. And I do not want to see a paper where half the paper or more is in quotes, and then you have a citation for where you got the information from. Okay, so most of the paper should be written in your own words, But if ever you want to use somebody else's words, and that's more than 3 of their words, you should put it in quotes and then cite where you got it from. And you use an in-text citation. I'll discuss that in a minute. Okay? Any question about any of that? The paper should have in-text citations, not only for where you're quoting someone, but you give an in-text citation whenever you're giving information that isn't common knowledge. For example, if you were to make a statement saying COVID-19 kills people when they get infected by it, that's common knowledge, so you don't need to have an in-text citation. But if you were to give a statement saying the number of people who died as of July 12th, 2022 in the United States is, it's something over 800,000 people in the United States have died of COVID-19. When you give a number that is a fact that is not common knowledge, then you have to give an in-text citation. Okay? Any question about any of that? So the paper should have in-text citations. You should have learned how to do that when you took the plagiarism tutorial, but students seem to have problems with that. So let me go and show you how to do an in-text citation. You'll have your text. I'm just going to go XXX for your text, and then you give your facts. Okay, XXX. And then at the end of the sentence, you put parentheses, and then the author's last name, Smith, and then the year, 2019. That's an in-text citation, okay? If the author is more than one, then you go Smith and Jones, meaning if it's 2 people, 2019. If it's more than 2, then you go the first author's last name, Smith et al., and then you give the year. We'll go a different year this time, 2013. Okay, that's how you do an in-text citation. And then each in-text citation, which is in your text, you should have references, which are your formal citations, references. So this would be like about a page. And then in here, you should give all of your in-text citations, where you go Smith, and you don't go just Smith 2019, you go Smith, and we'll go AL 2019. And then you give the where this came from. If it's a journal of National Academy of Sciences. And then the volume number. That would be a good volume number. 66. And then the page numbers. 1001, 1003. That's how you do it for a, this is telling you how to do a complete full reference, okay? Oh, I've got something missing in here. And there that, it'd probably be here. Give the article name. There it is. And that's how you do it for a journal. If it's from a book, it's a little bit more complicated. It'll be similar but different. For a book, you do that, and you would have perhaps the chapter, name of the book, publisher of book, and if it has chapters, you also have to have the editor. Name of a publisher book. There's one more thing, pages, and then city of publication. Hopefully that's everything. Chapter, editor of the book, the name of the book, publisher of the book, city of publication. I got the year there. All right, I think that's correct. And then if it's a web page, it's very easy. I didn't quite get all of that, but we'll go with it. For a web page, you give that same information, the author, the year, the name of the article, and then the web address. And that, oops, don't have that spelled right. That you usually do something like HTTP. W, whatever, okay? I'll put that in parentheses because that's what the, we'll just go XXX. Because that's the web address. Any questions about how you do a reference or in-text citation? I'll put that right here. This is something that students often get marked down on. That's why I'm taking the time to show you how to do an in-text citation. References, students tend to do okay. One mistake they do is you can't abbreviate the authors. If there's more than one, you have to list all of them. The exception is if there's more than 7, you have to list 7, and then you can use et al. Okay? But only if there's more than 7 can you ever use et al. In the full references. Any question about any of that? All right. The in-text citations are worth 10 points. You may use any commonly available citation format. I don't remember if you use APA or MLA in the plagiarism tutorial, but it discussed one of them. Just be consistent, meaning you only use one format. You don't do half with APA and then half with MLA. Do all one or the other way. Your paper on the infectious disease should cite the information that is appropriate, and you should have good citations. I think I require you to have 4 citations. I'm not seeing that here. Maybe it's down here. Yeah, you must have 4 unique citations that are good. Good citations are government websites, scientific journals like Nature Science, popular magazines, books and textbooks, newspapers from a credible source like the New York Times. You cannot use the National Enquirer. A variety of scientifically reputable television shows, websites, and other things like that. Generally, if the information is reviewed, then it is a good site. Okay. And for books, you can always look to see a reviewer. For journals, they'll state whether the article was reviewed or not, or whether the magazine or journal was reviewed. Okay. For web pages, it's hard to know if it's been reviewed or not. That's why you have to have a good, solid website if you're using that for a citation. Okay. Government websites, colleges are good unless you get material that is made by a student. All right. Be suspicious of anything that comes from a religious, a political, or a privately funded publication or website, because they will have their own viewpoint and they won't give you all of the facts. They'll skew it to what their viewpoint is. Okay. Personal websites, you can put up anything you want. It may not be factual. Colleges and university pages that contain student work should not be used. Wikipedia should not be used because anyone can go in and then change it. If you want to use Wikipedia, then you have to have a second reference to look up the information, meaning you can use Wikipedia, just realize you're going to have to get a second reference to, I don't know, prove that information. Any question about any of that? And Wikipedia usually gives references, so it's usually not a problem using Wikipedia and then using their references. Just make sure that they have references that work. You must do the literature citation page. You must have 4 unique sources that are good, and I can look at them and tell whether they're good or not. And that'll be worth 15 points. The report must be submitted in the folder in Canvas, and it must be either a Word doc or docx, a rich text format, or a PDF format. And don't go in and change the, what do you call that? The last initials of your document, because I won't be able to open it if you do that. So use the program that uses these, and then I'll be able to open it. I have this requirement to make sure that I can open the document and grade it. If you submit it in another format, I may not be able to open it, in which case you'll get a 0. All right, when you're doing the text of your paper, you should discuss the causative agent, and you should have a section heading that says causative agent. So if it's a bacterium that causes the disease, talk about the bacterium. Tell me its gram reaction, its cell shape, growth arrangement, and maybe some metabolic capabilities and other things that are unique to this organism causing the disease. If it's a virus, tell me if it's a DNA virus or an RNA virus. If it's an RNA virus, tell me if it's a positive strand or a negative strand RNA virus. Whether it's an envelope virus or a naked virus, how large the virus is, etc. So tell me about the causative agent. You should have a section. You should have a section on history. How long have we known about the disease? Historically, how was the disease treated? What's the history of the vaccine development of this disease? And describe or changes in knowledge and attitudes of the disease throughout history. Now, if it's something brand new like COVID-19, we haven't changed too much about our knowledge and attitudes. Okay, but we have changed a little bit about our knowledge about COVID-19. Like we know a lot more now than when it first came out. And our attitude has changed too. Nowadays, most people, they don't care if they get it and they'll live with it if they get it. They're not taking very many precautions. And so you can discuss it that way. Epidemiology, describe the prevalence and transmission of the disease, where in the world it's found, how many people are currently infected, what's the rate of new infections, morbidity and mortality, reservoirs, and the modes of transmission. Tell me about the pathology. Describe the pathologic effects on cells, tissues, and organ system. What tissues, organs, or cells does the pathogen affect? Like Staphylococcus aureus causing a skin disease, affects the skin. What damage does the pathogen inflict? How is the damage inflicted? Is a toxin produced? Does the immune respond? What is the time sequence of the disease? Like, tell me what the length of incubation time is. The prodrome, which we'll talk about what that is in a little bit, not today, but in a future lesson. The period of illness, the period of decline, the period of convalescence, and then what are the major signs and symptoms of the disease? Tell me about the response and the treatment, and then tell me about the social, political, and economic considerations of this disease. There is a copy of the grading rubric I will be using on Canvas, and it's right here. The government information will be graded as 1 point. The causative agent will be graded as 9 points. History, 10 points. Epidemiology, 10 points. Pathology, 10 points. Response and treatment, 10 points. Social, political, and economic issues, 10 points. The quality of writing, 15 points. In-text citations, 10 points. Literature citation, meaning the formal references, 15 points, for a total of 100 points. Any questions about this project? All right, pick a project and email it to me, and I'll get back to you about whether you can do it or not. Let's talk about lab 2, aseptic transfers. I don't have that anymore because I uploaded the lecture. It should be available to be seen now. Ah, lost it. Just a minute, I'm having trouble getting where I want. That's the exercises. There's the teacher copy. Let's also open the worksheet. All right. Lab 2. Aseptic transfers. Upon successful completion of this laboratory, students should be able to understand the purpose of using aseptic transfer techniques, determine if the aseptic transfer was used from the data provided, and be able to define the following terms: ubiquitous, aseptic transfer, inoculation, contaminant. There are some videos for you to watch in this lab. In our local environment, microorganisms are ubiquitous, meaning you'll find them almost everywhere. This is problematic when working with a microbiology lab because we generally want to work with pure cultures, a culture that contains one and only one species. In the mid-1800s, Louis Pasteur developed a technique for making aseptic transfers and minimizing the risk of culture contamination. And at about the same time, he also developed a technique for obtaining a pure culture. When you get contamination, you accidentally inoculated your culture with other organisms. And then you have a mixed bag of organisms growing in your culture. And the trouble with that is when you then get a result from a test, you don't know which organism caused the result. Okay, So we will generally work with pure cultures. You're going to learn how to make aseptic transfers. During inoculation, never lay the test tube cap or the Petri dish lid down on the workbench, because the workbench will not be totally sterile. Even if you sterilize it initially with the disinfectant, stuff can fall down on the workbench from the air. So always hold on to the test tube cap or the Petri dish lid while you're working on it. With the cap, you should hold it in one hand, and it'll be the hand that you have the tool that you're using to work with the organism. And then your other hand holds the tube that you're holding. Okay. For a Petri dish, you hold the lid over the area that you're working on, meaning over the dish, and then you work on the dish with the other hand using a tool. And that way, when an organism from the air falls down, most of the time it's going to hit the lid instead of going into your plate. Although contamination is possible, you're giving some protection. And then the tube, you hold it at an angle rather than straight up and down. Let me see if I can find something here. This cup will work. Okay, you have it straight up and down. Something can fall down. It's going to go into the tube. If you hold the tube at an angle, although things can fall into the tube, they're more likely going to hit the side of the container, in which case they don't contaminate your tube. Any question about any of that? And then you work efficiently and quickly so that you have the Petri dish lid off of the plate for as little time as possible, or you have the cap off the tube for as little time as possible. Meaning when you're not using it, put the lid or cap back on so it doesn't become contaminated. Another thing that helps is, of course, sterilize the area before you begin work, and always sterilize the area when you're done before you leave. In order to inoculate as new sterile media, inoculate is to transfer cells into, we must make sure that everything we use is sterilized beforehand. So the tube you get to inoculate should be sterile before you use it, but you do need to sterilize the tool you're going to work with. Because otherwise you'll contaminate your culture. And then these are some of the tools we use to work with, like a rack to hold up the test tubes, and then a Bacto incinerator for sterilizing our loop. And then we have a loop here for moving the bacteria from one location to another. Like if that's the media we want to inoculate, then this is where we have our cells. We sterilize the loop in the Bacto incinerator, cool the loop down, pick up the cells, and then move them into the new tube. Any question about any of that? All right, so here's a few terms you should know. And then we're going to do laboratory 2. You're not physically going to be performing any of these activities, but you will be watching videos on how to do them. We expect you will know and understand the material in these videos, as well as in this lab module. Procedure 1, you're going to transfer sterile liquid using a glass serological pipette. A pipette is a glass tool for moving liquids from one location to another, and we'll have a sterile pipette. In procedure 2, you're going to transfer a sterile paper disc using sterile forceps. So please watch the video demonstrating proper aseptic transfer by clicking on this link here. And let me see if that's in the worksheet. Yeah, it is. It's right there. Procedure 1, you're going to transfer 1 ml of sterile water into a tube containing nutrient broth. When you're moving things in a birette or a tube, you'll note that the liquid that you're measuring doesn't go flat across the container, meaning it forms a meniscus where it's, I don't know, convex shape. And where you read the meniscus is at the bottom of the meniscus. You do not read it at the top of the meniscus or halfway in between the meniscus, meaning the top and the bottom. Take the pipette and add 1 ml of sterile water into the tube of sterile media. That's what you're doing in procedure 1. And then we're going to let it grow and see if nothing grew, meaning you've performed aseptic technique. With procedure 2, you're going to use aseptic technique, transferring a sterile paper disc into nutrient broth. And we have sterile paper disc. You sterilize the forceps. I don't have any forceps, so I'll just use my fingers. How you do that is you put them in alcohol and then you pass the forceps through flame, or you could put the forceps in the Bacto incinerator. You can't get the forceps red hot because then you won't be able to hold on to it, but you can warm at least the edge of the forceps. All right. And it might not be perfectly sterile, but you will clean the forceps. Pick up the sterile paper disc and then put it into the tube of sterile broth and then incubate it overnight. And look to see if anything's growing in it, and if nothing's growing in it, then aseptic technique was performed. In procedure 3, you're going to transfer a bacteria, Serratia marcescens, into a broth culture. Remember, that's a liquid culture. In procedure 4, you're going to transfer the same bacteria, Serratia marcescens, onto an agar slam. That's a solid culture. For procedures 3 and 4, please watch the video demonstrating how to correctly transfer a bacterial stock culture to a sterile broth and to a sterile agar slam. Some important notes about this video. You only need to watch to 4 minutes and 23 seconds. The bacterium being transferred in this video is E. coli, and it will be a different color than Serratia marcescens. So that's something to note. And then there's an error made in the video. It happens between 3 minutes and 5 seconds and 3 minutes and 20 seconds. It's asking you, do you know what the error is? The hint is you can hear sizzling when the person puts the loop in the E. coli. And you should not hear sizzling, okay? All right, procedure 3, you're going to use aseptic technique to transfer Serratia marcescens into sterile broth. Serratia marcescens is pink in broth. It's red in a solid media. So it's a colored bacteria. Broth cultures are problematic because it is difficult to determine if contamination occurred because all the cells were mixed together. But you can determine whether there is a major contamination with Serratia marsans, because Serratia marsans in liquid broth or broth culture will be pink in color. And if there's major contamination, it will not be pink in color. Okay, if there's minor contamination, you won't catch it when it's growing in a broth, meaning it'll still be pink and you won't know. You should appropriately label everything you work with in the lab, and it should have your name, the date, the type of medium it is. This would be similar to NAY. NAY is nutrient agar with yeast extract. Well, this isn't an agar, it's a broth, so it's NBYE, nutrient broth, yeast extract. And then incubate it. The incubation temperature can be optional, and you must state what you have growing, and that you could say like air cultures, or in this case, you'd say Serratia marsans, because that's what will be growing here. And then incubate that overnight, and then take a look at it. This one, if sterile aseptic technique happens, you will get growth in the tube because we're putting bacteria into the tube. And then in procedure 4, you're doing the same thing. The difference is you're adding the bacteria to a slant. Here's a solid slant here, and you'll be streaking the bacteria in an S motion across the surface. Contamination is more easily detected in a slant or on a solid culture than in a broth, because if any contamination occurs, it won't be red in color like Serratia marsans. You'll have a region that'll be another color or off-white or unpigmented someplace on the tube. Remember to appropriately label this with your name, the date, the type of medium, and the inoculum. And then the incubation temperature you're growing at is optional. The interpretation of the results, use this table. For procedures 1 and 2, use this. The result on the left, the conclusion on the right. And you will have to write this in scientific terms where you're discussing the result and the conclusion. And then you need to say whether aseptic transfer was successful or whether it was unsuccessful. For procedure 3, you use the table here for nutrient broth. And then the conclusion here. And then the result for a nutrient broth, hazy or cloudy. Aseptic transfer was successful. And then the procedure 4, the results on the left, the conclusions on the right. And you need to use this table to answer the questions. Any question about any of that? All right. So you do have a question 0, which I think on most of yours, I've actually got that labeled. Oh, where's the worksheet here? Yep, it's labeled as 0. You need to answer that question. Describe the initial clarity of the nutrient broth prior to inoculation, before you added the sterile water. And you need to state, was it clear or turbid? And we have a picture of it. Right here. Image for question 0. For procedure 1, we have a picture of it right here. That's procedure 1. For procedure 2, we have a picture of it right here. For procedure 3, we have a picture of it right here. For procedure 4, we have a picture right here. use these results to answer your question and determine your conclusions, meaning this would be the result, how it looks, and then give your interpretation. And then answer the questions. For procedure 1, describe the clarity of the nutrient broth after inoculation with sterile water. Was it clear or turbid? Based on the results, what conclusion can be drawn? So you should state the conclusions. Was aseptic transfer successful or was it unsuccessful? And you do need to state it in scientific terms. List 3 sources of error with aseptic transfer technique that could explain why the nutrient broth growth showed growth of microbes in the tube. So you need to think about that. What are 3 possible errors that could have happened, which would cause the results that are seen? All right, for result 2, procedure 2, describe the clarity of the nutrient broth after inoculating with the sterile paper disc of the overnight incubation. State whether it's clear or turbid, and that's right here. Based on the results, what conclusions can be drawn? Give your conclusions. For procedure 3, describe the clarity of the nutrient broth after inoculating with Serratia marsans and overnight incubation. Was it clear or turbid? Give your conclusions. And how do you know whether you have transferred the correct microbe, which is Serratia marsans in this case? So answer that question. For procedure 4, it's very similar. Is there bacterial growth on the slant? Just answer yes or no. How do you know? When you look at it, how can you tell? And does it appear to be the bacterium you inoculated, or does it appear to be a contaminant? So answer that question. And then that's it for the lab. Type up your answers, submit it, and turn it in by this Saturday, 11:59 p.m. And then I'll grade it. Any questions? All right, if not, I will be here to answer any questions till 9 o'clock or the last student logs out of the class. It's currently 5 minutes before 8. So I'll be here for about an hour or until the last student logs off. Once the last student logs off, I'm going to log off as well. Any questions about what you're doing? If not, go ahead and get started. Let me state a reminder, and that is the lecture did continue for 10 minutes after I first said we were going to stop the lecture. I never stopped because I had the wrong time for when the class was ending. So I went on and lectured for 10 more minutes. If you're one of the students, I think there was one student who logged out, you need to see that lecture on YouTube. Thank you.